It sure is a beautiful day here at the Schiller Coastal Study Center. But wait, what could Sam and Maddy be looking for in the intertidal? Of course, should have guessed it. They're looking for green crabs. You may be familiar with this invasive species from your walks on the beach, but what you may not know is that it's really, really bad. There's been a lot of hoopla about these crabs destroying ecosystems. Fishermen complain about them eating bait, and environmentalists complain about them eating everything. Clams, eelgrass, mussels, snails, fish, lobsters, dogs. Seriously, everything. To tell the truth, we aren't that certain what they're eating. How do we know whether or not any of these claims are true? That's right, Maddy, we turn to science. There are many ways of attempting to figure out what's in those crab guts. Some old-school methods include visual investigation, where scientists try to identify hard parts like bones or undigested bits in stomachs.
I can't see anything!
There's always the good old-fashioned taste test. If we just want to figure out where these crabs are in the food chain, we can use stable isotope analysis.
But what if I want to know what they're actually eating?
Well, if you must know exactly what they're eating, radical new genomic techniques can help you find the answer. To begin the process, scientists carefully extract the stomach contents of crabs and isolate genomic DNA. By working carefully in a molecular biology lab, scientists can create millions of copies of the genetic information found in these crab guts. Genetic samples can then be sent off to special companies that will return the actual DNA sequences found within those samples.
Oh boy, I'm so excited for Illumina Sequencing to have called me to let me know my sequences are in. I'll go blast them right now.
Sam and Maddy can now search their returned sequences against every genome ever published. To do this, they use a basic local alignment test, or BLAST for short. It's as easy as copying and pasting a sequence into a website, or you can do it on a high-performance computing platform. Regardless of exact technique, the BLAST returns the sources of every strand of DNA extracted from those crab guts. So for instance, if the crab's been eating skate, we'd expect to find skate DNA. Looks like we've got some seaweed, some mussels, no surprises there. But wait a minute, what are cows doing there? It turns out that these techniques, generally called metabarcoding, may have their flaws as well. And scientists are working to overcome those flaws. But for the time being, genomics and metabarcoding are getting us closer to understanding what creatures eat and when than ever before.